My name is Justin Blethrow. I'm a product manager at PacBio. I mainly work on instrumentation. And it's my privilege and honor to host our workshop today. As I mentioned before, we have a lot of excellent speakers today. And we're going to hear a lot of stories of how our customers are using our technology to push the frontiers of genomics. But before we do that, I'd like to take just a few minutes to tell you about where things are at PacBio, particularly in our product space, where we've gone in 2018 and where we see ourselves going in 2019. So if we look back at 2018, one of our principal focus has been the end-to-end improvement of our product ecosystem to make it easier for you to use, to make it simpler and more successful, and to produce more data at better economics. So your projects are more affordable, they're more capable. We've made an effort to do this all the way across the workflow, from the library prep side to the sequencing consumables, to the instrument itself, and to the downstream analysis software. And I want to take a few minutes to highlight some of the places where we have made changes there. On the library prep side, one of the key things we did in 2018 was to introduce the SMRTBELL Express Template Prep Kit for very large insert libraries of 30 KB and up. And this is a much improved, streamlined, additive workflow that allowed you to take a process that was a day and a half on average and take it down to about 4 hours of hands-on time, simultaneously decreasing the requirements for input DNA and producing less DNA damage in your sample at the end of the process. On the consumable side, the sequencing consumable side, we had a couple of chemistry releases actually, the most significant of which was the 3.0 chemistry, which we released just a few months ago now really, which contained a number of improvements, but very notably it contained a faster polymerase. At the same time, we released a version 3 of the SMRTCELL 1M, which contained a wide variety of improvements to the chip itself in its production process and a variety of things like that, that afforded greater reproducibility and deterministic behavior in loading of the cell. One of the challenges we've faced in the past before this was more variability from one run to another than we'd like. The SMRTCELL 1MV3 really took us a long way down the road in terms of reducing that variability. On the instrument side, we made a lot of software changes both to the primary analysis, the signal processing that goes on inside that translates the chip data into actual sequence. And we made a lot of improvements on the reliability and reproducibility front. It's a complex system. There's a robot involved, a lot of workflow events. And we had a very concerted effort, actually, mainly in the first half of the year, with several focus teams involved to go through and find any source of variability or unreliability we could, and we prioritized remedying those in the software releases this past year. And I think by and large we were very successful, and as I'll show you later on in some field data, I think it's contributed greatly to improving the reproducibility of our yields. Lastly, but by no means least, on the analysis side, we had an important release on the SmartLink software, which included improvements for some of our key applications, notably structural variant calling, improvements to ISO-SEQ, and we also added new tools on the RunQC side and in barcoding analysis to help you better understand your sample and how it perhaps could have been optimized or where you might want to go in the future with your experiments. We also made HGAP compatible with Falcon on Zip to enable our customers to combine the benefits of those two. So where is the system today in terms of raw performance? I'm gonna show you results in two categories, for very large inserts, and then I'm gonna show you the results we're seeing today for shorter inserts. This is an example of a well-loaded, well-prepared, high-quality sample run in-house, where we're achieving 15 gigabases of yield. This is a 35 kilobase size-selected human library. And you can see we're getting upwards of 500,000 reads and a polymerase mean read length of just under 30,000 here. Half of the data is in reads above just under 44K. we've been able to, at the same time that we've been able to increase the yield from these cells, we've been able to maintain and actually improve the quality of the raw reads themselves. We've pushed with the 3.0 chemistry, we've pushed our average accuracy up towards 91% in some cases. And when we look at how that translates into consensus accuracy, we see that we're achieving QV50 at 40-fold coverage, QV40 at about 20-fold coverage. Now, I mentioned I'd just make a distinction between long insert and short insert libraries. If we look at what happened in 2018 on the shorter insert libraries, shorter in PacBio terms, 10 KB still being quite long by a lot of metrics, there's something really interesting that happened. If you look at this run yielded 43 gigabases, which is about 4 times higher than we were a year, 18 months ago. This is from just over 500,000 reads, and the average read lengths are quite long at 84,000 mean read length, meaning that half of the data is in reads greater than 167,000 base pairs. So looking retrospectively for a minute, you know, we've seen, I think we've seen significant yield gains from the system in both the very long insert and these more medium insert applications. But something sort of intriguing and profound has been happening, in my opinion, with the smaller inserts. So if we, as I was commenting to before, if we look back a year or so, we might have expected to get about 10 gigabases from one of these runs. And with the chemistry release early in last year, we take that up to maybe 20 for a really well-run experiment. And now we're seeing runs that can achieve up to 50. So what is going on here? How did we achieve this, and what are the implications? There are a lot of things that go into making this possible, but one of the key aspects is that the 3.0 polymerase that we released with the 6.0 system software is extremely processive. A lot of engineering work went into making this happen, and we managed to obtain a polymerase that will just keep going. So this has really profound implications when we think through how this translates into some of our applications. It's not really an evolutionary improvement. It turns out it's actually quite revolutionary. So circular consensus sequence is something that PacBio used to talk about some, and then was less prominent for some years. And then now with these recent chemistry gains, it's very much back in the spotlight. And I want to take just a few minutes to explain what it is for any of you who aren't familiar. So this is a cartoon of a polymerase circling around a relatively short template. Because we have hairpin adapters on each end, the polymerase is able to sequence the first strand, then the second, and then go into rolling circle mode and just keeps chugging out sequence. If that template doesn't have any physical limitations itself, any DNA damage, and you have a very processive polymerase, you can get extremely long reads here. When you think about the long insert case shown here, just statistically, those inserts are more likely to have some kind of limitation inherent to them, like DNA damage, given an equivalent quality of input DNA and equivalent care and handling, just statistically, because they're longer. So these long insert reads, while we've seen very significant gains, going from 5 gigs per experiment up to now 15 and sometimes even higher, we haven't seen this sort of 5-fold improvement that we're seeing on the CCS side. There's another thing we can do afforded by the tenacity of this polymerase, which is something called pre-extension. And this is simply that we can start the sequencing in the dark before we turn on the laser and add the complication of possible photo damage from the dye. And there's a neat trick that comes from this. In that we can load the cell more highly than you'd actually want it to end up being, and rely on a sort of natural selection process for any templates that do have damage to go ahead and die off before we actually start sequencing. So by the time the actual sequencing run kicks off, you have a target population of really healthy templates that are in rolling circle mode, and they will just give you long reads. Now, when you produce those very long reads, you end up with multiple independent observations of the exact same molecular sequence. And because our error is highly random, when you combine these multiple observations in software, you end up washing out the vast majority of error. And so we can get long reads, 10, 15, in some cases 20 kb in length, with extremely high accuracy levels, the kind of accuracy you come to expect from NGS type sequencing. And this has allowed us now to bring structural variation analysis, which has been one of our fortes, and single nucleotide variation into the same experiment. The graph I'm showing you on the right is a representative of how many passes you need to achieve a given quality level with the 3.0 chemistry. And perhaps you can see that with just 4 passes, a read on average will have about 99% accuracy, or QV20. And with 10 passes, you get all the way up to Q30 at 99.9. And if you think back on the slide I showed you where the N50 on the data was 167,000, it's not hard to get 10 passes on a very large template. Importantly, in doing this, we have preserved some of the key aspects of our sequencing capabilities. For example, the independence from GC bias. This is an experiment looking at the coverage by GC fraction bin in the human genome. And we see that there's essentially no bias as a function of the GC content. So looking again at this example experiment, we did use an 8-hour pre-extension in this case, and we loaded high, and that allowed us to end up with over 500,000 ZMWs actively producing high-quality sequence in the end. And on processing, this translated into nearly 300,000 reads at Q20, or above Q20. Actually, many of them were far above Q20. If you're interested in learning more about this kind of approach and the exciting things we think it can do, I'd point you to this paper led by our own Aaron Wenger and a number of other scientists at PacBio, and done in collaboration with Google and other parties. It's just gone up on bioRxiv. It's a very exciting paper. I'm nowhere near bioinformatically capable enough to do it justice myself, but I would strongly encourage you to take a look. So turning now for a moment to what we're seeing customers do in the field. It's one thing for me to stand here and make claims about what kind of yields you're getting. It's another to look at what your peers might be achieving. So this is a graph of customer data anonymized, but where we have the yield numbers per run tracked over time. And I've highlighted the 2018 period here at the bottom. You can see that both with the 5.1 launch and with the 6.0 launch, there was a strong upward trend in the yield per run that customers have been achieving. It's important also to note that when we do make one of these releases, the fleet doesn't immediately convert. It takes some months for customers to adopt it. So you would expect a damped effect from these upgrades, and yet we see a fairly sharp transition in both cases, which tells me that it has indeed had a very strong effect. And if you look at 2018 as the whole, and bearing in mind that this is an average of all kinds of experiments, very short reads, very long reads, we see that the average yield has more than doubled in 2018, going from somewhere around 5 to close to 12. If we look a little closer at the field performance by system release level, there are a number of interesting additional trends that we can see. So these are in chronological order, essentially from left to right, and you can see the population evolving towards higher yields. But perhaps more importantly, or equally importantly, the very left side tail, the low yielding cells that in the 0 to 2 and 4 gigabase bins have plummeted. And this is reflective of some of the variability and reproducibility improvements I commented to earlier in the talk. We've eliminated a lot of sources of outright failure or underperformance or very low performance. There's some residual even in the 6.0 that's, I think, reflective at this point mainly of properties of the sample, not exclusively properties of the sample. We still have occasionally underperforming runs that are our fault, but we've managed to eliminate most of that now. Another interesting thing to note in this is that the population is, if we focus just on the 6.0 population, It's really two distinct subpopulations at this point. That we have the pseudo-Gaussian on the left, which represents customers doing very large insert experiments. And in that population, you can see that the mode is around 12 gigabases. And then there's a very long tail out towards the right. And these, we've looked a little more carefully. These are shorter inserts, customers doing CCS, doing ISO-SEQ kind of runs. And you can see that it tails well into 30s, even in the 40s, and all the way out to 50. But if we throw out the fact that this is two populations for a moment and just look at the overall statistics, the average output per cell these days is 13 gigs on the 6.0 release, and 96% of runs produce 5 gigs or greater. And now only about 4.3% of runs produce what we would consider a very low performing yield of less than 3 gigs. It's been gratifying to see that our customers are observing this performance increase firsthand, and a lot of them have commented about it on Twitter. Mark Gasser, for example, had a tweet mentioning back-to-back days with 18 gigabases per smart cell. Genomics had some ISO-SEQ runs in the low 40s per smart cell. And more on application focus, Kevin McKernan mentioned achieving the highest quality by far cannabis genome to date using this new chemistry. Sorry, one moment. So we're particularly gratified to see these system-level performance improvements translated by our customers into application-level performance improvements across our key applications in the plant and animal science space. For example, our customers are able to target more and more complex genomes, large polyploid plants, for example. They're able to achieve higher accuracy, great accuracy in representation of haplotypes. And in some cases, and perhaps we'll hear more about this very shortly, they're able to expand the scope of their experiments to look at related species or multiple exemplars within a species to really start understanding diversity and relatedness. We're also seeing much broader adoption of ISO-SEQ as it has become easier to use and more performant, and customers are really beginning to understand the ability for this approach to accurately characterize transcript diversity. We've also had some interesting results applying an approach that was initially focused for human genomics, at least internally, looking at the ability to characterize and discover structural variants now in plants. And here we have a couple examples where our PBSV software was applied and our data was applied to maize and soy, and we achieved far greater detection of structural variants than was possible with NGS technologies. So looking back over the whole arc of PacBio's process, our product development process, we've increased the output per smart cell over 2,000-fold, which is gratifying, but where are we going to go next? I think 2019 is going to bring about some very profound, arguably transformational capabilities in our product ecosystem. And again, I'm going to call out some highlights within each aspect of the product portfolio. On the library prep side, we're going to continue to make this less burdensome for you, make it easier, faster, higher quality output. At the same time, we're looking to add new capabilities, such as the ability to generate high-quality reference assemblies on individual organisms from individuals from small-bodied species. On the sequencing consumables and instrument side, we are going to be releasing the SMRTCELL 8M that you all are probably very curious about, as well as a Sequel II instrument to support it. On the analysis side, we will be releasing SMRTLink 7.0. This will continue to have applications improvements, speed up new capabilities such as copy number variant detection in the structural variant application. But also very importantly, it's going to have a lot of improvements in the efficiency of the algorithms, just the time to results, as we seek to support instruments that are now generating 8 times more data per unit time. Right? That's a very challenging cliff to adopt or to face with your existing infrastructure. So we're doing everything we can to make that transition easier, and so that you can get further along with your existing compute. If you'd like to hear more about the details of that upcoming SmartLink release, I'd encourage you to attend the workshop on Wednesday that I'll discuss a little bit more at the end of the talk. So turning back just for a moment to the sample, or to the library prep side of things, we're going to make the template express, SMRTBELL Express template prep kit that we introduced in 2018 now general purpose. You may recall I mentioned that the original version was limited to insert sizes of 30 KB and above. We've eliminated that limitation, and it's now suitable for all library sizes. It retains all the benefits of the original version. It's a streamlined protocol. It has about 4 hours of hands-on time. And because it's a single tube additive workflow, it minimizes effort and risk to your sample. And as a consequence, also, it has lower gDNA input requirements, around 3 micrograms, which we expect should help simplify upstream protocol requirements going into it. This will be available sometime in Q1 2019. We haven't finalized the release date on this yet. We're also very excited about the upcoming low DNA input protocol, and I don't want to steal too much thunder on this because we're going to hear more about this a little later. But the goal here was to enable you to achieve high-quality de novo assemblies from individual small organisms, flies, worms, other species, where getting enough material in a clonal sample is really challenging, and people have had to come up with rather burdensome compensatory approaches. This is going to be a roughly 4-hour prep. It relies on a modified version of the Express template kit 2.0 that I just described. There's no DNA shearing or size selection involved, and it ends up using about 100 to 200 nanograms per experiment to achieve these results. Genomes up to about 1 gigabase, given high quality DNA, can be covered in this manner. And we have a few examples shown in the graph where mosquito and fly and worms all achieved pretty impressive contag N50s with very low inputs. This was developed in collaboration with Mara Lanichak, I hope I pronounced her name correctly, and Matt Berriman at the Wellcome Sanger Institute, and we're grateful for their assistance. We're also making improvements to the ISO-SEQ technology, both on the bench side and in the analysis software. Liz is going to tell us more about this, so I'm not going to go into great detail. But suffice to say, we've endeavored to make it, and succeeded in making it, faster on both sides. The workflow has been streamlined, and then on the analysis side, the algorithms have been made much more efficient, so the time to results are much lower. At the same time, we've increased the analytical power of it. We've improved the accuracy. We've improved the number of full-length transcripts that are coming out of the prep. And we're very excited for where that will take us. So turning now to perhaps the thing everybody's most eager to hear about, the 8M and the Sequel II instruments. So this will be coming. I'll talk about the schedule a little bit more in a minute. And it's going to enable our customers to achieve much more sequence data per run. And in particular, it's going to make, as well as supporting scaling of these long insert applications. We're very excited about the synergy between the scaling and the kind of polymerase read length behavior I described earlier, and what this means for our ability to do long reads at high accuracy on a large scale affordably. So in terms of raw performance, this is a glimpse at where the system is today. This is a pair of runs from a human 35K size selected library. We ran it on the 1M sequel for reference and achieved about 15 gigabases with a little over 500,000 reads. When we run it on the 8M cell, we achieved just under 100 gigabases per run, 96 in one case and 99 in the other. Read lengths were slightly shorter on the 8M case than in the 1M, but this is most likely due to loading differences and the fact that we have not fully optimized. This is a pre-release chemistry, and we're still updating and making changes here. We've seen similar results in additional species. Here's an E. coli example coming in at 86 gigs, a B. subtilis example at 116 gigabases, and rice coming in at 85. These are all 10-hour runs, importantly to note. So in terms of gigabases per hour, I don't think anything out there comes close. Importantly, we've achieved, or we've maintained our ability to deliver high consensus accuracy in these runs, and we can achieve QV50 or above with about 40-fold coverage. So now looking at the performance with shorter inserts. Again, 10 to 12K inserts, in this case from human. And taking long movies, because the polymerase can just keep going and going. We wanted to see what we could get. In this case, on the 1M, we got about 44 gigabases, very much in line with the data I showed you earlier. On the 8M case, we got over 300. We got 320 in one case and 318 in the other. And I'm not convinced that the loading has been optimized here. I think we could go higher as we fine-tune the system. In one case, we had 4 million reads, slightly over 4 million reads. The other were just under that. And the average read lengths continue to be comparably long to what we're seeing with the 1M system. Importantly, this can translate into quite a large amount of high accuracy long read data. So we're looking at a histogram of the post-circular consensus processing data on the bottom right by read quality and number of reads. And you can see that the apex of that histogram is just under QV40. And the strong, maybe half of the data is above 30. The average over the entire population is 99.8%, so just under QV30. And in terms of yield, this boils down to 21 gigabases of data at this quality. So we're very excited about what this means. In the long insert case, if you think about a 1 gig genome, one run is going to give you 90-fold coverage. If you're taking the CCS approach, again, one run is going to give you 21-fold coverage. And given the high accuracy at long read length of that data, we think that's going to be very powerful. Just to show the equivalence of the chemistry as it stands today, the 0.9 pre-release chemistry on the 8M versus the 1M chip, we're seeing really comparable behavior in terms of how the number of passes translates into a read quality score. And in fact, we're seeing marginally better behavior on the 8M today. Not so much that I'd say that this is a solid trend. It could just be experimental difference. But in one case, we're achieving QV20 at about 4.5-fold. And on the 8M case, we're at 3. So we're quite pleased with the performance of the system and the opportunities we think it will provide our customers. Right now, the system is an early access program. It is in the hands of some of our customers who have just begun to kick off runs. And we expect this process to run about 8 to 12 weeks. We have certain objectives in mind for it, and there's a little bit of flexibility to account for unforeseen events, infrastructure down, that kind of thing. But we expect it to run about, maybe, I'm going to say 12 weeks. And then the commercial release is expected to follow reasonably shortly thereafter. We want to take enough time to process the things we learned from the early access program and reflect that into the product. So it's difficult for me to commit to exactly how far after it's going to be. But we're very eager to get this product out to the market, so it's not going to be much time. So lastly, I just want to comment or remind people of a few activities and events going on here. We are doing our Smart Grant program again this year. The program has not kicked off yet, but the website is live if you want to go and take a look and familiarize yourself with it. If you want to participate and win smart sequencing for a project of your interest, you need to submit a 90-second video abstract between February 1st and 22nd. And you can learn more about this at the website indicated there. And we're very pleased to thank our co-sponsor, Histogenetics, on this. As I mentioned before, there is a Smart Informatics Developers Conference on Wednesday following the majority of PAG. It starts Wednesday at noon and lunch will be served. If you're interested, the details are here, or contact any of the PacBio personnel and we can make sure you have what you need to attend. You'll hear all about ongoing informatics developments and upcoming things in SmartLink. Lastly, we are conducting a fun raffle at the booth where we're giving away 4 cells worth of sequencing and bioinformatics support for the data that comes from them. And if you're interested in participating in this, please just drop by the booth and you can get all the details. Many of you already have, I think.